1, and we are live. All right, first thing I wanted to look at and point out to you was here. This is right from the protein synthesis section in your chapter 10. And this is a great picture to show you exactly what's going on as you've gone through your notes now, where each of these two processes is taking place. So again, we're looking at that central dogma. We talked about that DNA is housed in the nucleus. And again, how do we get those instructions out to the ribosomes to make the proteins? So the first process is called transcription. And again, we'll go into each of these processes more in detail next week. But again, just a good general overview is transcription is taking place in the nucleus. And again, you're going to unwind the DNA to get a section of a gene that's going to code for a specific protein. And you're doing that by using messenger RNA, which again, we see the table over on the board, right? RNA is single-stranded. So now that small segment of a single strand of messenger RNA is small enough to come through the nuclear pore and then get out to the ribosome, where you then go through the second part of translation to build that protein. And proteins are built from amino acids. Amino acids, or a protein can be anywhere from hundreds to a couple of thousand amino acids long. It just depends on the type of protein. All right, so that's just a general overview of what's going on there. And again, we'll go into a little detail next week of what's going on here and what's going on here. But we want to start actually practicing. So let's look at this one here. So this one's a little more detailed. We're going to get to this point next week. But again, if you have your DNA code, we're going to go through this whole process and determine which amino acid these three are going to code for. So if you have DNA here, and I've got a code, I've got an adenine, a thymine, and a cytosine. What would be my complement to this? Just go to this. What would pair with A? T, is that correct? What would pair with T? A. What would pair with C? G. Okay? Because again, you've got your DNA code and we've got the complement DNA, so our rules still hold. A goes with T, C goes with G. Let's complete it. What would be this? C, C, G. And then what would pair here? G, G, A. Right? So that all makes sense. That's nothing new. Okay, but now we're going through this transcription. So we're taking this DNA code, because the DNA has now been unwound, because we're going to start to build a protein. So we're going to code this into our mRNA, which is called the codon. So again, that's the beginning part of transcription. So remember, this is mRNA. So we no longer have thymine, we have uracil. So A would pair with what? U. T would pair with A. C. Actually, we're going to go ahead and reverse this. All right, just for this example here, we're going to go ahead. All right, let's go ahead and look at it again. So A goes with, T goes with, C goes with, G. All right, now we're going to go ahead and take this side, and then we're going to transcribe it into mRNA. So T goes with, A goes with, and then G goes with, so AUC there. All right, now over here, what would we have? And then what would we have here? Ooh, nice. Right. So the other thing too is what we'll get into. This mRNA is actually going to code for your amino acid. But as we'll find out when we get to translation, in order for that amino acid to be linked onto the protein, you have to have what's called an anti-codon. And again, this is all chemicals we're talking about. So it's like a check. It's a check to make sure you have the correct amino acid. So we'll see that mechanism when we get there. But again, A would pair with U would pair with, C would pair with, G, G, C, and then over here would be, that's a C, right? So what do we need? A. Okay. So now what we can do is we can take, we're going to take these codons now that we have our check, and then we're going to determine which amino acid that is. If you looked in the book, I don't know if in the video, did she talk about the codon chart? Yeah, a little bit. So you have your codon chart here. So this was the old school where you would take your first base, your second base, and your third base. But then over time, they've developed the codon wheel, which is very nice to work with. So automatically your eyes are drawn where? Where? To the center, right? To the exact center where the letters are the largest. So that's where you're going to start. So if your first letter is, say, an A, then you would move out to that second ring. And then depending on if, let's say you were an A again. And then if you were, so now you see this A is going to break out into the 4. And then let's say you were an A again. So you would go A, and that would be lysine. And really all you're looking at is the 3 letters there. So even though it's a little crooked, I like this one because it's actually giving you the full name of all the amino acids, and it's giving you the 3-letter code. Like, I'm not making you memorize any of these amino acids, but you just want to be familiar with the 3-letter codes. And you also want to be careful because some of them are a little similar, like where you've got ASN, ARG, ALA, ASP. The other thing too is you've got amino acids that have, I think, up to 6 different codons, right? So you've got leucine here for these 2, you've got leucine here for these 4, and then I'll draw your attention to a couple other ones. Methionine is the one that is usually the start. So again, in some of the examples we give you, just because we're trying to practice, you might not always have a start and a stop. But when you're building a protein, in order to initiate it chemically, you're going to start with this AUG, which is methionine. And that's considered the start. Now you start building a protein, and then you're going to continue that process until you get to one of these 3 stops. So you would have UGA would be a stop. You would have UAA as a stop, and then UAG is a stop. Right? Does everybody seem a little comfortable? Like you start here, then you focus on your second letter, and then you go to your third letter. So if we have AUC, that would be isoleucine, so it would be ILE. So then you would write down here ILE. Let's move to the second one, GGC. GGC is glycine, so it's GLY. So then we put our GLY. And then our last one, oh, what the heck? It just broke my pen. You see that? My knowledge is so powerful, I can break physical objects. That's not true. All right, so CCU, let's check that bad dog right here. So we got CCU. So we go right here, CCU, and that's proline. So that's PRO. So we go over there, and we have PRO. So that's the whole big one. You'll have some of this practice next week. What you're going to start off slowly is with this one here, where you're really just going to 3 steps. You're going from the DNA that's going to be transcribed straight into your codon, and then you're just going to go ahead and look up your protein. All right. One thing I would recommend, just that I do automatically just for housekeeping, is I just break, because again, a codon is going to have 3 letters. I just break them up. So if I see your paper like this, that's awesome, because then you just keep track. You don't have to lose anything there. So again, if you're going from DNA to mRNA, A would pair with what? U, T pairs with, and then G pairs with. So UAC, let's see what that is. UAC is tyrosine, so that's TYR. So that goes here. So then you would put TYR. All right, so just quickly again, going back to your codon wheel, and I think we did talk about this back when we looked at proteins, when we looked at macromolecules. Again, they're going to code for these 20 amino acids, which run your body. These amino acids build the protein. We even talked about, I think your body makes 11. The others you have to get through your diet. So again, going in order, you got asparagine, lysine, serine, arginine, valine, alanine, aspartic acid, glutamic acid, glycine, phenylalanine, leucine, serine, tyrosine. You got your stop, your cysteine, stop, tryptophan, leucine, proline, histidine, glutamine, arginine, isoleucine, methionine, threonine, asparagine, lysine, serine. All right, so those are those. But again, you're really just concentrating on that. I think that hopefully is everything you need to know right now. So again, we got I love questions. Yes. Yes. Oh, so again, going back here. So again, this is all done chemically. So again, how does your body know when to stop building on a protein? It's going to come up with that chemical, and it's going to come in the shape of either a UAA, a UAG, or a UGA. And again, that's just a sequence of your DNA. So again, that's the gene. You're going to have the methionine that's going to start the process of building that specific protein, and then you have a stop, one of the 3 stops. So again, if your protein is 1,200 amino acids long, okay, that first one is methionine, and then when it gets to that 1,200th, which is one of these 3 stops, then it knows that it's good to go. Then it can build that protein. And again, this is all done chemically. Oh, yes, correct. So if you wind up with that code, it stops. So that's the only one that's really not a 3-letter digit. So you would write MET if you got the start, and then you would just write stop for any of the 3 codes there. But some of this practice it's not gonna be strict, because it's just trying to get you to, get to all the different proteins. So really what you have then is you got 1, 2, 3, 4, and 5. You have until tomorrow at 3 to do that. I'll give you an assignment tomorrow that it starts to look at transcription. But really, you want to get this to me so I can look at it over the weekend to see if we have any issues. And this will be a simpler one, and then next week we'll come back and look at something like that. And as always, I talk way too long, but that's okay.